It's now my great pleasure to introduce our final speaker of today, Dr. Coral Zhou. So Coral did her undergraduate in biochemistry at the University of Maryland before moving to UCSF for her PhD studying chromatin modelers with Geeta Narlikar. For her postdoc, Coral then moved to Berkeley to work with Rebecca Held, where she has been studying mechanisms of mitotic chromosome scaling in xenophus. And Coral has been very successful at every stage of her career with great publications and numerous awards, including the Jane Coffin Child Memorial Postdoc Fellowship. And she has also made important contributions to justice, equity, diversity, and inclusion initiatives, as well as in outreach and community service efforts. All that to say, I'm thrilled we're able to have Coral speak to us today on her research on the mechanisms of genome size control during embryogenesis and evolution. So please join me in giving a warm, albeit virtual, welcome to our final speaker.
Thank you so much, Ben, and for all the organizers for giving me this chance to talk to a chromatin audience, which is always fun for me because my old crew. So let me share this screen. Okay. Great. Okay, so today I'm going to tell you how we can use these really cute frogs up here to study really fundamental mechanisms about how our genomes are shaped and sized over time and space. And so I'll start by just emphasizing this idea that I think many of us don't think about, is that size is actually this really fundamental feature across biology. And when we study biological scaling, we actually really want to answer two big questions. So we want to know, what are the causes and consequences of a change in size? And we also want to know, what are the evolutionary constraints on size? And so to illustrate these concepts, we can take an example from organismal biology. So on the left here, I'm showing you a plot of body weight versus skeleton weight for a really large range of organisms. And what you can hopefully appreciate is that these two parameters have this really beautiful linear relationship. And this may seem really intuitive, but we can actually learn something more meaningful if we dig deeper. So if we now just look at the metacarpal bone from a smaller animal versus a larger related animal, we can also see that bone size now scales to skeleton size. And since we know the material properties of bone itself, from these dimensions, we can actually calculate a bone compressive strength, which mathematically relates to a bone load, which is really the maximum amount of weight that this bone can withstand without breaking. And then what we find is that across species, and including in humans, this bone load is always 10 times greater than body weight. And so we think this scaling factor of 10 is really what evolutionarily constrains the ratio between bone size and body size. And of course, the function of this scaling relationship is so that when an animal is walking or running, its body doesn't get crushed by the weight of its own bones, or its bones don't get crushed by the weight of its own body. Right. And so something that I've been really fascinated by is, can we actually apply the same exact framework to studying genomes? And I think we can. So if we look at genome size across the tree of life, I'm showing you here in this plot, in terms of the number of base pairs of DNA per genome, we can actually see that there's this huge range, right? So between the smallest bacterial genome all the way over here to the largest plant genome over here, there's about 6 orders of magnitude in difference. But what I would argue is that, unlike that bone example that I just showed you, we really don't have a good fundamental understanding of how genome organization and genome function really scale with genome size. And we really care about this question from sort of like an intellectual and also from a basic biology perspective. But we really also care from a disease perspective, because cancer genomes are known to be abnormally large. And in fact, nuclear size has been used really commonly as a prognostic marker for many cancers. So here I'm showing you tissue samples from patients with breast cancer. And I hope you can appreciate that on this right-hand sample, where these tissues have bigger nuclei, that actually correlates with a worse prognosis, right? And so we really would like to know from also this disease perspective, how is it that genome size scales with genome organization and genome function? Okay, so if we wanted to ask this question, there's a couple of ways we can really do this. And one maybe obvious way is we can take different species with different genome sizes and we can ask, how are these parameters down here changing? But that becomes a little complicated because these genomes themselves, in terms of DNA sequence, are really different. So there's actually a simpler way to ask this question, and that's within a species and within a single genome, we can ask, instead of the amount of DNA or the content of the DNA, we can ask, how does that genome fit into a certain physical space, and how does that change over time? And we know that actually a time when this is actually changing a lot is during early embryogenesis. So in the lab, we love these Xenopus laevis frogs because they can lay a ton of eggs overnight. These eggs are humongous. They're over 1 millimeter across. And we can actually go ahead and fertilize these eggs in dishes. And upon fertilization, the embryo undergoes these rapid and successive cell division events, as shown here in this video, resulting in smaller and smaller cells over time. And we also refer to this stage of development as the blastula stages. And I'm showing you this video for a frog embryo, but in fact, this general process is really well conserved among all metazoans. And so over the years, people have used immunofluorescence, for example, to really look inside of these embryos. And what they have seen is that subcellular structures, such as the mitotic spindle, shown here, as well as just nuclei, actually scale with cell size during this time, and they get smaller. And so over the past 20 or so years, my lab, among others, have really figured out the fundamental molecular mechanisms for how these sizes are differentially set as a function of cell size. But what we really didn't understand was the mitotic chromosomes, which of course sit at the center of these mitotic spindles, really ready to be segregated into daughter cells. We didn't understand how they behaved. And in fact, some older work had shown that mitotic chromosomes also become more compact and shrink over developmental time, which I'm showing you here. But some of the open questions when I started my postdoc were really, when is this scaling starting to happen? And also, how is it that mitotic chromosome size is being set as a function of developmental stage? And these are some questions that I really solved and answered in this publication that you can access online. And so I'm just going to give you some brief highlights from it because it's been published. I mainly want to sort of end the talk by telling you about what I've been up to and the main question that I really want to explore in the future, which is why. Like, what is the function of all of this scaling? Is there even a function? What can we learn from this? So I'm just, like I said, going to share with you just a few highlights. So I'm going to go ahead and just summarize a bunch of in vivo data in the paper and say what I was able to show is that these chromosomes are actually scaling during these blastula stages. In fact, even during the 4-cell stage, they're starting to scale. And by the time they've exited the blastula stages, they've shrunk about twofold, primarily in length and not in width, okay? But I really wanted to understand what is the molecular mechanism behind this, right? Which is really hard to do in vivo for a lot of reasons. And so instead we turned to the extract system. So because these frogs lay so many eggs and because these eggs are so big, we can actually go ahead and just collect these eggs in a test tube and do a really simple fractionation step, in which case you separate out the cytoplasmic layer, which is this golden layer in the middle right here, away from sort of the egg-specific factors, the energy stores of the egg, so lipids up here and yolk down here. And this cytoplasm we call extract, and you can think of extract as really just undiluted cytoplasm. It has everything that you would find in the cytoplasm. It has mitochondria, ER, Golgi, all of these cool things, but it's in a test tube, right? So we really love this approach because it's kind of this in vitro, in the middle of in vivo and in vitro, where you can do all of these cool things to extracts that you could really not do in vivo, or at least it would be really, really hard. And so the egg extract is really cool specifically for two reasons. So one is that, in fact, because of the pelleting step, the extract actually does not have a genome because that maternal spindle that's in the egg is pelleted away. So we can actually add back our own source of genome. But the other cool thing about egg extract is that even though it's biochemically arrested at metaphase, you can actually really easily transition it between metaphase and interphase. And so to mimic early blastula stage of development, what I do in the lab is I'll add purified sperm nuclei to interphase egg extracts, in which case the sperm will decondense, the chromatin will replicate and recruit full nuclear envelopes. And then I'll add back fresh metaphase extracts, in which case the chromatin will now form these condensed mitotic chromosomes, as well as these bipolar spindles. And once I get a tube of spindles like this, I'll dilute the extract with a little bit of fixative and do a really quick purification of the chromosomes through a glycerol gradient and onto cover slips. And now I hope you can see that these chromosomes are really beautifully individualized, and I can go ahead and measure their lengths and do things like IF. And this is exactly the kind of morphology that we would never really be able to get in vivo. Okay, so then to mimic sort of a late blastula stage of development, what I do is I'll grow up dishes of these embryos. I'll arrest the embryos in G2, and then I'll go ahead and make a G2 arrested stage 8 embryo extract. And then I'll pellet those nuclei and resuspend that pellet in metaphase arrested egg extracts. In this case, we also get bipolar spindles, but now in the middle of these spindles are actually stage 8 mitotic chromosomes. So when I go ahead and compare these two sources of nuclei and the kind of chromosomes they make, I again see that there's this twofold difference in chromosome length. And it's not just the full difference, but in terms of the absolute values of these chromosome lengths, which are plotted on the right, we can fully recapitulate the chromosome dimensions that we had measured in vivo. And this was really exciting to us because what it suggested is that these egg extracts are actually able to recapitulate mitotic chromosome scaling, but in a test tube, in which case we can do a lot of different perturbations. And so using this egg extract system, what I went ahead and did is asked a couple of really basic questions. I wanted to know, first of all, what are some of the biochemical factors that determine chromosome size? So again, we use a lot of the single chromosome IF, in which case I'm always normalizing to a DNA stain to really calculate the abundance of a certain factor on the chromosome. And the second question really was, what are some of the architectural changes that occur along these chromosomes as they shrink? And so for this, we turned to our genomics approach using Hi-C and polymer modeling. And this was all through a collaboration with the Decker lab. Okay, so when it came to trying to identify potential scaling factors for mitotic chromosomes, we immediately turned our attention to two really important enzymes, condensin 1 and topo 2, which are each necessary, but together actually sufficient to build mitotic chromosomes out of chromatin. And these two enzymes really sit dynamically on the central scaffold, which you can see here in cartoon form, but also based on an IF stain of condensin 1 on a human chromosome. And essentially the idea is that condensin 1 in particular can form these DNA loops that kind of emanate outward from the chromosome. And the idea is that the morphology of these loops should then contribute to the morphology of the whole chromosome. And then the third factor we became really interested in was linker histone H1, which sits dynamically at this entry-exit site of the nucleosome, because our lab had shown many years ago that if you deplete H1 from egg extracts, you actually get longer mitotic chromosomes. So I went ahead and measured the abundances of these three factors on the short embryo chromosomes versus the long sperm chromosomes. And I found that these short chromosomes actually have less condensin 1, less topo 2, but more histone H1. And I'm actually going to go ahead and skip the Hi-C data since it's a short talk and go straight to the model. And so we think that based on both pieces of data, that these short chromosomes actually have longer DNA loops, as well as more DNA density per layer of the chromosome as you go up the axis. And this kind of explains how you can accommodate the same amount of DNA, but on a shorter chromosome axis. And at the same time, these short chromosomes have less condensin 1 and less topo 2, and at the nucleosome level, they have increased histone H1, which could lead to increased nucleosome compaction. And so what's really cool about this model is that even though we've known these three factors are important for mitotic chromosome assembly, now what I've been able to show is that during this physiologically relevant process of chromosome scaling in the embryo, how these three factors can really come together to build a twofold shorter chromosome. Okay, so a really big part of my project has been trying to understand how is it that these different abundances of these scaling factors are actually being regulated through developmental time. And so I'll just give away the punchline. What I found was that there are two factors, two developmental cues that independently set chromosome size in the embryo. So one is cell size that decreases over time, and the other is this nuclear cytoplasmic ratio, which actually increases over time. And that's what I'm going to talk about today. So the way that I'm defining nuclear cytoplasmic ratio is really the number of genome copies per volume of cytoplasm. And so because there's no cell growth during the blastula stages, every time there's a cell division event, you actually get an increase in NC ratio. And it's thought that, and as a result of this increase in NC ratio, the abundance of certain maternal factors per genome actually goes down over time. And this sort of titration effect is really thought to be one of the main triggers of what activates the zygotic genome for transcription at the end of the blastula stages. Okay, so I went ahead and just asked a really simple question. I wanted to know, is NC ratio actually sufficient to recapitulate scaling? And so all I did was I again turned to the egg extract system and I varied the concentration of sperm nuclei that I was putting into egg extract by 16-fold, which really mirrors sort of an early versus late blastulus stage of development. And so then what I found was that, in fact, simply by changing the NC ratio in the test tube, I was able to decrease chromosome size by about 1.5-fold. And in fact, this actually really beautifully mirrors the in vivo data. And so we think, in fact, NC ratio is sufficient to perform this scaling. And at the same time, what I found was that these shorter chromosomes actually do recruit less condensin 1, which is really consistent with my previous IF results. And I'm not showing the data here, but we actually got less significant results for topo2 and condensin 1. Okay, and then the real surprise came when a wonderful undergrad in the lab repeated these experiments and looked at nuclear size and spindle size. And what she saw was completely different. She saw that nuclear size pretty much stayed the same, and she saw that spindle size actually increased. And I want to emphasize that these are, of course, not the effects that we would expect in vivo in the embryo. And so our interpretation of this is that while NC ratio is sufficient to scale mitotic chromosomes, there are other overriding cues in the embryo itself that actually set the sizes of these other two structures. Okay, so then I wanted to know, when during the cell cycle is this NC ratio Q actually being set? Is it during interphase or is it during mitosis? And so to answer this question, what I did was repeated these NC ratio experiments with two nuclear sources that actually essentially get rid of interphase. So the first is by using stage 8 embryo nuclei, which again have already completed their interphase in the embryo. And the other was to use unreplicated sperm nuclei. So instead of allowing the nuclei to replicate in the egg extract, in interphase egg extract, we can actually just directly add these sperm nuclei to metaphase egg extract, in which case they'll still form mitotic chromatids, but they're completely unreplicated. So what I found was that in either case, in fact, there was no longer a relationship between NC ratio and mitotic chromosome size. And so when we go back to this model, what we think is happening is that, in fact, this NC ratio Q is being set during interphase. And we actually don't really know what that Q is yet. I tried pretty hard to figure out, but that's something for the future. But whatever it is, it's being set during interphase, and then it's being read out during metaphase through decreased condensin 1 loading over developmental time. Okay, so I'm going to skip over the cell size data because it's all published, and I just want to summarize everything into a model. And so what we think is going on is that these mitotic chromosomes are shrinking during these blastula stages through about a 2-fold change in chromosome length. And we think that these shorter chromosomes have less condensin 1, which allows them to create these bigger DNA loops, which is again how we accommodate the same amount of DNA, but on a shorter chromosome axis. And we also think that at this nucleosome level, that the increased amount of H1 on these shorter chromosomes can also lead to increased internucleosomal compaction. And that's something that we're actually actively testing by using cryo-ET and FibSem to look at the nucleosome packing in native samples. And so really a key feature of this model is that these two developmental cues are actually completely distinct from one another, both in terms of temporal regulation, but also in terms of the effector molecules. So I didn't get a chance to show you all of this data, but NC ratio is exclusively set during interphase and only through this condensin 1 pathway, whereas cell size is being set during metaphase and exclusively through the H1 pathway. Okay, so I started this talk by kind of introducing you guys to this idea of size and biology and in terms of how we think bone size scales to body size. And now what I've shown you is how we think mitotic chromosome size also scales to cell size and NC ratio during early development. But what I really haven't told you at all is why any of this is happening or if it's even important. And so there was a model that was proposed a while ago that suggested that maybe you actually have to scale mitotic chromosome size to spindle size in order to prevent chromosome missegregation. But this model has never really been tested. And so now that we really have the molecular knowledge of how to perturb mitotic chromosome size as well as spindle size, we can kind of do that independently or in conjunction in the embryo and actually really test this model. And so that's one of the future directions that I have planned. And the other thing is maybe the reason why you need to set these different sizes within the spindle is actually, it has something to do with the physics of how these chromosomes segregate. And maybe having a difference in chromosome size actually changes the biophysical properties of the chromosome. And so this is something that I'm also actively testing in collaboration with John Marco's lab at Northwestern. In a couple of weeks, I'll be going there to stretch these chromosomes and really measure sort of the underlying biomechanical properties of these different sized chromosomes to fit into this model. Okay, and so we can also test these ideas on an evolutionary timescale by using different frogs. And so we have this other very cute frog in the lab. It's called Xenopus tropicalis. It has a smaller genome, a smaller body size. It lays smaller eggs, and it makes smaller spindles in vitro. And so the question becomes, are these scaling relationships that we've already determined in Laevis, are they consistent in tropicalis, or are they broken, and why? And what we're really pumped about is that now we're just starting to expand our arsenal of different frogs in the lab to include these other two species that have really huge genomes. So there's a dodecaploid and dodecaploid here, and also an octoploid over here. And so we're really curious, and I'm working with Gabe in the lab on this, to assemble their transcriptomes as well as their genomes. We're really curious how their genomes have really adapted over time in terms of how they've adapted to size. And so by assembling these transcriptomes and genomes, what we're really hoping to understand is sort of at the DNA sequence levels, for example, the coding sequence of different histone chaperones or different chromatin packaging proteins, how those have really changed over time. And we can actually test those mechanisms in vitro as well. And this is very much a team effort. So Gabe and I have a lot of help from two wonderful junior technicians in the lab, John and Martin. And these tadpoles are about 2 months old now. So these are Longipes tadpoles. And it's a lot of work to do all the husbandry, but hopefully once they get to adults, we can really send these samples out for sequencing. And this is also, we're not bioinformaticians, so I also want to acknowledge our wonderful collaborators, Adam Session and Taejun Kwan, who are really doing the ones putting together these transcriptomes and genomes. Okay, so as a graduate student in Guido Narlikar's lab, I was really fascinated by these chromatin remodeling motors, and I specifically studied Inno80, which is this amazing multi-subunit like machine that grabs these nucleosomes and really twists them in a way to really precisely slide a nucleosome. And one of the things that I found during my research is how modular these enzymes are. And so going forward, I'm really excited to be able to combine my knowledge and expertise as a chromatin biochemist, as well as now a cell biologist, to ask this really fundamental question of how is it that genome organization and genome function really scale with size across all of these different units of chromatin that I've highlighted in red. And I think I might just skip the last part of the talk because otherwise I won't have time for questions. But anyway, the general approach is that I'm going to be able to combine sort of the in vitro systems that I learned in Gita's lab with sort of these extract and also in vivo systems that I've learned in Rebecca's lab to really dissect these mechanisms, but then also test them in vivo in the embryo. So I'm going to skip this and go straight to, acknowledgements. So I want to thank, of course, my wonderful collaborators whose work I didn't actually have time to show today, Job and Bastian Decker. They were really fun to work with on this project. Some other collaborators that I've been working with, some lovely frog folks for antibodies, some help from illustrators, and also some image analysis experts, my lab, and my wonderful advisor, Rebecca, who really taught me a lot about cell biology, and these two wonderful students who worked with me on this project. You can check out the paper, it's out, and you can always contact me, and of course funding. So happy to take any questions. Thank you.
Thank you, Carl. That was a beautiful talk. I really enjoyed that. I was wondering if you kind of talked about this a bit, but if you could expand on like for the varying genome sizes,
yeah,
and something like the spindles, like how do you see the spindles having to evolve in response to different genome sizes? Or is that a way that you're thinking about this?
Yeah, actually, that's exactly Gabe's plan. Gabe is really a spindle expert, and he wants to know. So what he's doing is actually he's taking nuclei from one species of frog and putting them into extracts from other species to see if he can essentially break the spindle. And he's seeing some really crazy stuff with like the Longipes nuclei, putting them into Trop or Laevis extracts. The spindles get really, they don't form these nice bipolar structures anymore. And then he wants to figure out the molecular mechanism behind that using the sequencing approaches. And so I'm more interested in the genome packaging side of things, right?
Yeah.
So, but yeah, that's something the lab is thinking about.
And have you thought about looking at other, like from the point of view of the animal, like other points where the size of the genome might matter? Like I know like people have talked about like in mammals versus birds, like red blood cells, for instance, which enucleate or not, and how genome size could matter there. Have you thought about looking at other points in biology where the size of the genome might or may not be constrained?
Yeah, definitely. So I think one of these aspects is in cancer. I think that cancer cells kind of have their own rules for how they can evolve, right? So they a big event that happens in cancer cells is whole genome duplication. And this is something that's not really tolerated very well in most normal cells. It still happens normally in some cell types, but not all. And so the thought is that the cancer cells want to duplicate their genomes several times over so that they can actually then harbor these other deleterious mutations that would normally not be tolerated and would normally kill the cell. But people don't really understand how that works. And so what we love about the frog system is that these whole genome duplications have actually happened really naturally over evolutionary time. And so we can kind of learn about how the frog genomes have adapted to these big changes in size. And then maybe there will be some correlation with what happens in cancer. Yeah.
Great. Yeah, I'm still just, my mind's blown with thinking about the 12N frog.
Yeah. Yeah.
So Shubham has his hand raised, so go ahead and ask your question.
Thank you very much. That was a really, really great talk. Please correct me if I'm saying it wrong, but you mentioned that the ratio is set during the interphase and then it's read out during the metaphase, right?
Yeah.
Two of the players you mentioned are Condensin and Topo2A. So we know that Topo2A is preferentially recruited to sites where the DNA is supercoiled. And there is at least 2 studies now showing that condensin is also preferentially recruited at the tip of plectonemes, which are formed in supercoiled DNA. So I was thinking if the transcriptional differences at interphase actually dictate how much of condensin or how much of topo2 you are gonna actually recruit, and that then that would show up. So I was wondering if you have looked at transcriptional differences at the interface between the 2 scenarios, because that could be one possible mechanism for how you see a difference in condensin versus topo2A recruitment, which then drives the behavior that you see.
Yeah, absolutely. I really love that idea and of thinking about DNA topology. So one thing I failed to mention is that actually in the stages that I'm looking at in development, there actually is no transcription. So transcription actually gets activated sort of at the end of the blastula stages.
I see.
But something that I have noticed is that when I've tried to add sort of transcriptionally active nuclei into egg extracts to make these mitotic chromosomes, I actually get this huge failure in formation of chromosomes. And I've never really dug into why, but it's always been in the back of my mind that maybe there's some kind of incompatibility between the biochemical factors in egg extract and whatever chromatin, maybe topology or organization that's already being created because of transcription in the embryo. And so I think that could be a really fascinating direction. And maybe it does have to do with topology like you're suggesting. Yeah.
So just a quick follow-up. So you mentioned that you see a failure. So is that a failure with forming like the mitotic spindle?
No, they form spindles fine, but when you look at the mitotic chromosomes themselves, they're really poorly formed. They're not individualized. They aren't these beautiful rod-shaped chromosomes that I would normally get in the sort of transcriptionally silent samples. Yeah, but I could do things where I could try to inhibit transcription in the embryo and then make the nuclei that way and see if that makes a difference. Yeah, those are things that maybe I'll plan for the future. Yeah.
Yeah, and just maybe I can ask one more. So what's your hypothesis regarding why you see differential recruitment of condensin Maybe you talked about it, but I missed it. Like, do you have a hypothesis in mind that you think would be playing a role?
Yeah, maybe I'll go back to that model. So the idea is that, so my hypothesis for a long time was that it was H3. And so the idea here is that, that as you increase NC ratio, you just have more and more genomes, right? And so
Right.
then the abundance of certain maternal factors per genome actually goes down over time, right? And so the question really is, what is that factor that's going down over time, that then is decreasing the amount of condensin 1 loading that's happening?
Right.
And my hypothesis was that it was going to be histone H3 loading, because people have shown that histone H3 loading goes down in nuclei over time. But then I found in my work that there's actually no correlation between histone H3 abundance and condensin 1 abundance, actually. So I don't think it's H3. It's got to be something else. Yeah.
I see.
Mm-hmm.
Awesome. Thank you very much.
And then Irene said she had a question, so you can ask it.
So I had a question about your planned future work comparing the different frogs. You mentioned you wanted to look for protein coding changes, so it's sort of a two-piece question. One is, have you looked at the scaling factors you mentioned in other genes whose proteins might be interacting them across species whose genomes you already do have, to see if there are actually differences between species that are related to genome size? And a related question is, do you think that most of the differences you'll see will be in protein coding regions, or that these regions are potentially very highly conserved and there might be some regulatory region differences that are affecting their expression levels that are actually causing these chromosome size differences?
So all of the above, Irene, I would say. So right now we're just starting to get the transcriptomes, and we're going to do some targeted searches for things like condensin, for things like spindle scaling factors, to really compare across all 4 species. But I'm being told that we actually want the genomes as well, right, in order to look at these non-coding things, but also in order to really faithfully know that, you know, this sequence is coming from this chromosome, and there's so many different isoforms and things like that. So we're really interested in all of those things you're suggesting. So thank you.
Thanks for a great talk.
Thanks.
And then we are slightly over time, but if you have a minute, there's a question in the Q&A that.
Okay.
So this is from Duraj Joshi, who says, great work. Are the protein/mRNA of condensin and H1 inherited? And is the association of H1 and condensin and the chromosomes dependent on the dilution of the inherited levels of condensin or accumulation of H1?
Yes, definitely. Great question. So yes, all of the proteins and also mRNAs are inherited from the mother, and they're all stored in the egg in really high amounts. So I don't actually know for condensin 1. My assumption is that condensin 1 is not. It's just the protein that's being titrated away over time. I have read that histone H1, even though there's a great storage of it in the egg, it's also being translated over developmental time during these blastula stages when there's no transcription, but there's active translation. And so one of the experiments I can do is also see what happens to scaling if I inhibit translation. And so I haven't done that yet.
Awesome. Okay, I think we'll wrap up there. Thank you again for the great talk. And thank you to our other 3 speakers as well. I thought this was I really enjoyed the session. Thank you for everyone who attended. We'll be back again in 2 weeks with the normal seminar. So, bye everyone.